Blueberries boost longevity beyond calorie restriction. This comes from the Life Extension magazine. Cellular DNA endures relentless injury through internal and external factors. A fundamental problem with aging is faulty DNA repair. As we lose our ability to repair DNA, illness and death ensues. If we could perfect internal DNA repair processes, we would become incredibly resistant to degenerative disease. Blueberry extracts have demonstrated robust DNA repair properties. The memory-boosting and brain-protecting benefits of blueberries have long been a promising area of research. Newer studies are showing that blueberries delay the aging process through a variety of mechanisms, including repairing DNA damage and favorably modulating genes associated with aging. The ability to repair DNA is an essential longevity mechanism. A fascinating laboratory study found the compounds in blueberries could increase mean lifespan by 28%. In humans, that is equal to over 22 years. Particularly impressive is a study indicating that when added to a calorie-restricted diet, the compounds found in blueberries significantly increase lifespan beyond that of calorie restriction alone. All of this adds up to a powerhouse fruit that could help us live longer and healthier. When scientists began investigating blueberries, they first discovered the fruit's impact on reducing the age-accelerating effects of oxidative stress on tissues. Blueberries are unique among fruits with their ability to not only quench destructive oxygen-free radicals, but also to directly modulate genes that impart cells with their own protective mechanisms. Those genes produce enzymes such as superoxide dismutase and catalase that endow cells with the ability to capture and detoxify free radicals, producing harmless water molecules instead. Blueberry constituents do much more than fight oxidation. Studies are now revealing the powerful ability of blueberry polyphenols to modulate genes associated with aging itself, such as the Methuselah gene, which can promote an extension of up to 35% in the lifespan of fruit flies, a commonly studied organism in aging science. Studies in no fewer than 3 different species illustrate the value of blueberries in decelerating the aging process to produce significantly longer lifespans. The simplest multicellular organism used in aging research is the roundworm C. elegans, a minute worm measuring about 1.3 millimeters long, which is one of the most versatile animal models of aging. Studies reveal that treating C. elegans worms with blueberry polyphenols increase the animal's mean lifespan by 28%. In human terms, that's the equivalent of adding more than 22 years to the life of a 79-year-old person, which is the mean life expectancy of a male child born today. C. elegans worms are extremely sensitive to temperature variations, and blueberry treatment increased the animal's survival during acute heat stress. Treated worms also were much better able than non-treated animals to resist induced aging stresses. Indeed, blueberry treatments can prolong the life of the classic laboratory organism, the insect Drosophila melanogaster, better known as the fruit fly. Flies fed on blueberry extracts lived on average 10% longer than did control animals. This increase was accompanied by an increase of multiple beneficial genes, including protective enzymes and reduction of age-shortening genes. These healthy effects were apparent when flies were treated with an herbicide called paraquat, which normally imposes fatal age-accelerating changes in the fly's metabolisms. Of course, roundworms and insects are far removed from human beings, and studies show that benefits in such animals must be reproduced in higher organisms in order to be applicable to humans. That has recently been done in laboratory mice. In one study, the polyphenols from blueberries, pomegranate, and green tea extracts were combined into a nutrient cocktail added to mouse chow, and the effects on the animal's lifespan and biochemistry were examined. Middle-aged mice were placed on one of 3 dietary patterns. Control mice were allowed to eat as much as they wanted. An intermittent feeding group was fed only every other day to simulate the life-extending properties of calorie restriction. A third group received the polyphenol mixture in addition to calorie restriction. As expected, both the calorie-restricted and the calorie-restricted plus polyphenol groups significantly outlived the control animals. What was remarkable, however, was that the calorie-restricted group that also was fed the nutrient polyphenols significantly outlived those on calorie restriction alone. This demonstrated the additive effect of the polyphenol mixture onto the known life-extending effect of calorie restriction. The brains of the polyphenol supplemented animals showed reduction in the expressions of deleterious life-shortening genes. What makes this study so noteworthy is that the longevity benefits of calorie restriction are so robust that it is difficult to improve them. Yet adding extracts from blueberry, pomegranate, and green tea enabled lifespan extension in a mammal greater than calorie restriction alone. Of course, in this study, it's difficult to discern which of the extracts, whether blueberry, pomegranate, or green tea, may have been responsible for the observed life-extending effects. All three are rich in the anthocyanins that individually extend the lifespans of lower laboratory animals. They also have specific effects that counteract age-related diseases that contribute to shortening lifespans. So, how do blueberries manage to prolong life and improve function so dramatically? We're nowhere near a definitive answer given the complexity of the berry's biochemistry, but some key points are beginning to emerge centered on the body's own ability to repair itself. Blueberries and their polyphenol components appear to be exceptionally good at promoting repair of DNA strands and at optimizing immunity at the cellular level. Both of these functions are essential for preventing senescent and environmental impacts that lead to cancer, cardiovascular disease, and loss of metabolic control that underlies obesity and diabetes. DNA, the molecular blueprint that contains instructions for all life functions, is a delicate molecule that, despite many protective mechanisms, readily suffers damage from oxidation, radiation, toxic chemicals, and other sources of stress. All living cells have evolved mechanisms to identify and repair broken DNA strands, which, if left unrepaired, promote cancer and early cell death. Numerous studies now reveal that blueberry extracts both prevent DNA damage and promote its rapid and accurate repair. In an impressive human study, healthy volunteers ingested either 300 grams of ground blueberries or a controlled jelly with no blueberry components. Blood samples were taken before, in 1, 2, and 24 hours after the subjects took the study supplement. At the end of 1 hour, the amount of oxidation-induced DNA damage in blueberry supplemented volunteers fell by 18% compared with control subjects. What makes this study particularly impressive is that this single, albeit large, serving of blueberries was able to suppress this amount of DNA damage so quickly. Ultraviolet light produces DNA damage that plays an important role in causing a variety of diseases. When scientists expose cells in culture first to UV light and then to blueberry extracts, they found both a reduction in reactive oxygen species and reduced DNA damage. Closer examination showed that cells were protected even after DNA damage had occurred, the mechanism being related to decreased genetic expression of stress-induced signaling proteins. This study showed that blueberry anthocyanins protect DNA from damage and protect cells from the consequences of DNA damage that did occur. The actions of many immune system components decline with advancing age in a degenerative process called immunosenescence. Controlling and slowing immune senescence is an attractive means of prolonging life and health. Blueberry extracts have been shown to have powerful anti-aging impacts on both cellular and molecular components of the aging immune system. At the cellular level, blueberry extracts boosted the actions of natural killer NK cells charged with identifying and destroying abnormal cells, such as those infected by viruses and those showing malignant changes. At the molecular level, blueberry anthocyanins sharply decreased activation of nuclear factor kappa beta, which is an inflammation-generating molecule associated with cancer, heart disease, and other manifestations of chronic inflammation. Successful longevity requires more than just DNA repair and slowing immunosenescence. Increasing cardiac health and reducing the risk of cancer and diabetes are also part of the equation. Blueberry compounds play an active role in reducing these risks as well. Increasing one's intake of berry polyphenols is a proven means of protecting the heart, blood vessels, and brain against cardiovascular diseases such as heart attack and stroke, which remain among the top causes of death in American adults. Blueberries contain one of the highest concentrations of such polyphenols among common fruits, and these compounds have been shown to inhibit the dangerous oxidation of low-density lipoproteins, LDL, the so-called bad cholesterol. Lipid oxidation constitutes one of the first steps in developing plaques that grow and become cholesterol-filled, inflammatory lesions that ultimately block blood vessels. In rats, a year-long diet regimen containing 2% blueberry supplement following experimentally induced heart attacks, myocardial infarction or MI, produced by tying off one of the heart's main arteries, was able to completely arrest expansion of the damaged area of heart tissue. Expansion of the MI area is a leading cause of the heart failure that so often follows a heart attack and produces much of the death and disability we associate with heart attacks. In this study, the death rate over the 1 year of the study was 22% lower in the supplemented animals compared with the control group. Blueberry supplemented animals' hearts showed less of the deleterious remodeling of the heart chambers that is also a hallmark of heart attack damage in humans. Another seminal study in rats showed that a 3-month blueberry supplemented diet produced a 24% increase in a measure of the resistance of energy-releasing heart mitochondria to stress. Remarkably, in animals then given a heart attack by tying off their coronary arteries, the size of the infarction, the dead area, in the heart was 22% less in the supplemented than in the control animals, while the number of inflammatory cells was reduced by an impressive 40%. When supplemented rats were put back on a regular diet, devoid of the blueberry extract, their infarcted dead heart lesions grew. When control rats were put on the blueberry supplement, their infarctions stopped expanding. This study indicates that if one suffers a blockage to their coronary artery when taking blueberry extract, the damage to their heart muscle will be lessened. If one suffers heart muscle damage from a coronary blockage, damage to their heart muscle might be halted if they initiate blueberry supplementation. Blueberries and their polyphenols relax blood vessels, which in turn reduces blood pressure and may help to slow atherosclerosis. Human studies demonstrate reductions in both systolic, top, and diastolic, bottom, blood pressures. A decrease in systolic blood pressure is observed when animals are fed blueberry supplemented diets, even when the diets are very high in fat. These effects could be related to blueberry polyphenol-induced modulation of nitric oxide, the signaling molecule that blood vessels use to relay information about blood flow and pressure, and to shift mineral ions such as potassium across cell membranes. There's also growing evidence that blueberry polyphenols can induce favorable structural changes in blood vessel wall composition. This results in arteries becoming more elastic and less stiff, further reducing blood pressure and lowering atherosclerosis risk. These kinds of favorable vascular changes have been shown in human studies when blueberry polyphenols were administered to healthy people. When a fairly high dose of 766 milligrams total blueberry polyphenol intake was given to human subjects, blood vessels smoothly dilated within an hour of the dose. Another study showed that in sedentary adults, a blueberry supplement produced reductions in aortic systolic blood pressure and in aortic stiffness. All of these changes are highly desirable for prevention of atherosclerosis and cardiovascular disease in general. Indeed, in humans, a sharp reduction in cardiovascular risk factors was shown in people with metabolic syndrome, the cluster of obesity, blood sugar intolerance, and lipid disturbances that tremendously increase risk of a heart attack or stroke. At the cellular level, blueberry extracts, including those of root, stem, leaf, and fruit, appear to improve glucose transport into cells, a critical mechanism for lowering blood sugar. Blueberry fruit extracts also enhance formation of new insulin-producing pancreatic beta cells, which would otherwise die off as a consequence of high glucose levels. Specifically, the The researchers measured the critical ability of cells in the pancreas to produce insulin in response to high glucose levels in the bloodstream. Scientists had discovered that high blood glucose levels damage insulin-producing cells in the pancreas over time. But if these same cells are treated with blueberry stem, leaf, and fruit extracts, the number of viable cells increases. This is a very desirable feature, especially for people with early type 2 diabetes, in whom some sensitivity to native insulin production still remains. Furthermore, the researchers tracked the source of the improved insulin response to a remarkable increase in replication of the remaining growth-arrested beta cells by nearly threefold. In addition, they found that blueberry extracts from stem, leaf, and fruit could prevent the glucose-induced toxicity and subsequent death of neuronal cells by up to 33%. This finding has tremendous implications for prevention of neurodegenerative diseases such as Alzheimer's, which has been called type 3 diabetes, to acknowledge the important role played by glucose toxicity. The benefits of these effects can be seen in a human study in which obese subjects, not yet diabetic but with proven insulin resistance, consumed either a smoothie containing 22.5 grams of blueberry bioactive molecules or a smoothie of equal nutritional value but without blueberries, twice daily for 6 weeks. In the blueberry group, insulin sensitivity, which is the ability to remove glucose from the blood in response to insulin, rose to more than fourfold that of the plain smoothie group, even without significant changes in adiposity or energy intake. Simply put, blueberry supplementation gave this high-risk group of patients vastly better natural control of their blood sugar. Other studies in animals reveal powerful effects of blueberries on the other components of metabolic syndrome as well. In obese rats on a high-fat diet, adding freeze-dried whole blueberries to the diet reduced not only blood sugar and insulin resistance, but also lowered abdominal fat mass and triglyceride levels in blood. With their powerful anti-inflammatory and gene-regulatory properties, blueberries seem an ideal agent for preventing cancer as well. Human studies in cancer prevention are enormously difficult and time-consuming, but there is ample preclinical data available to support an important role for blueberries in the prevention of malignancies. In addition to their unequaled content of bioactive anthocyanins and other polyphenols, blueberries are the single best source for pterostilbene, a close relative of resveratrol, which is also found in grape skins and other dark fruits. Pterostilbene, a compound found in blueberries, has been called a hallmark anti-cancer agent based on its ability to fight malignant change in several tissues while producing negligible toxicity. Along with pterostilbene, the anthocyanins in blueberries exert a spectrum of effects, all of which reduce the risk that a cell will become cancerous, and that an incipient cancer will grow, invade tissue, and metastasize to other body sites. Documented effects of blueberry bioactive molecules include prevention of DNA damage, arrest or alteration of the cell proliferation cycle to reduce the growth rate of cancerous cells, induction of the normal programmed cell death routine that is abnormal from malignant cells, allowing them to proliferate infinitely, and prevention of tissue invasion and inhibition of metastasis by downregulating protein-melting enzymes used by cancer cells to invade. Plants that live in challenging environments produce a wide array of stress-reducing nutrients to help them cope with extremes of temperature, humidity, nutrient availability, predators, and other threats. When humans consume those molecules, we get the benefit of that genetic stress resistance, which helps our bodies fight off major threats to our own well-being. Blueberries, which grow in harsh, sandy soils, often in dry conditions and at high altitudes, contain the highest known concentrations of many such bioactive molecules. Research shows that in addition to their ability to protect brain tissue and function from the ravages of aging, blueberries contribute to better health in most body systems. Blueberries and their extracts now show promise in preventing and mitigating human cardiovascular disease, metabolic syndrome, which includes obesity, lipid disturbances, and hypertension, and cancer. Daily supplementation with standardized blueberry extracts may help ward off life-shortening degenerative diseases currently treated with prescription drugs. The most exciting finding, however, is that blueberry, pomegranate, green tea extracts may enable you to live significantly longer, even if you're already practicing calorie restriction. You've been listening to an article originally published in the Life Extension Foundation magazine, April 2015, called Blueberries Boost Longevity Beyond Calorie Restriction. If you like the information you just heard, please support us by subscribing to this YouTube channel, but most importantly, by purchasing products at scotthealthsystems.com. We don't ask for donations or any paid subscriptions, but you can support us by purchasing our great products. Not only will you keep getting other great free informative videos about health research like what you've just heard, but you'll also get fantastic products like our Garcinia Cambogia Advanced Slim Factor Formula with 95% HCA and chromium. It's fantastic to prevent sugar from being converted into body fat, reduce appetite and cravings, and improve insulin response so that you burn your calories instead of storing them as body fat. It's the perfect weight loss formula to be taken before meals that contain starch or sugar. It's a powerful, safe, effective formula that contains 95% pure HCA, unlike many other companies. Please click on the link below. Thanks for listening, and please support us by visiting scotthealthsystems.com.